Alzheimer's disease is physically characterized by the accumulation of toxic amyloid-beta proteins. These proteins clump into jagged, invasive plaques that weave through the neural network, creating a physical barrier that suffocates the brain's ability to communicate. Healthy brains use adult neurogenesis, the birth of new neurons, to maintain and repair circuitry. But as the disease progresses, the neural stem cells responsible for this process slow their activity and eventually enter a dormant state. Previous attempts to combat this by artificially boosting neuron production had limited success. While new cells improved memory function slightly, the existing amyloid plaques remained untouched, leaving the toxic environment intact. This suggests that creating new neurons is only half of the solution. For a treatment to be effective, it must also address the biological waste that prevents those new cells from functioning. Researchers at Riken developed a genetic treatment called iPad to target these dormant stem cells. They tested the protocol on 5xAFD mice, a standard model for human Alzheimer's pathology. Following the treatment, the mice demonstrated a clear recovery in cognitive function. They regained the ability to navigate complex mazes and showed significant improvements in spatial memory tests. The data in these charts tracks the physical density of amyloid plaques over time. Note the trajectory of the iPad group in the red columns. The introduction of new neurons coincided with a quantifiable decrease in the area covered by toxic plaques. This presents a logical contradiction. A new neuron lacks the mechanical tools to dissolve protein plaques on its own, yet its presence triggered a broad reduction in toxic material throughout the surrounding tissue. This indicated that a larger cleanup operation had been initiated. To find the source of the clearance, the researchers began investigating the cellular interactions within the hippocampal tissue. Researchers identified a non-cell autonomous effect. A change in one cell type, the neural stem cell, triggers a functional shift in surrounding cells. The birth of new neurons sent signals to the brain's support network, astrocytes, and immune-focused microglia. Microglia serve as the brain's native cleanup crew. Once mobilized, they physically migrate toward the amyloid deposits to engulf and digest the plaques through phagocytosis. At the same time, astrocytes increase the production of lipoproteins, like ApoE. This secretion helps metabolize the amyloid protein, making it easier for the brain's waste disposal systems to flush it out. The newborn neurons effectively mobilize the existing cellular ecosystem, providing the biological signal needed for the astrocytes and microglia to begin the cleanup. Identifying the cells involved was only the first step. The next goal was to find the specific genetic regulator that initiated this system-wide response. To map this, researchers performed RNA sequencing on the hippocampus. This volcano plot visualizes the results, showing hundreds of genes altered by the iPad treatment. Among the suppressed genes, one emerged as a high-priority candidate, PRAG-Ka2. It was consistently downregulated during maximum plaque clearance. This gene is directly linked to human pathology. In post-mortem studies of human Alzheimer's patients, PRAG-Ka2 is found to be significantly overactive. Scientists tested this link by using a virus to knock down, or silence, the PRAG-Ka2 gene in the hippocampus, bypassing the need for stem cell rejuvenation entirely. The results confirmed the gene's role. On the left is the control tissue, heavily laden with amyloid plaques. On the right, the tissue where PRAG-Ka2 was silenced shows a dramatic reduction in plaque density, mirroring the results of the full iPad treatment. These results indicate that PRAG-DAG2 is a primary regulator of the brain's internal clearance mechanisms. Suppressing its activity appears to be a requirement for initiating a broad biological cleanup. Lowering the activity of PRAG-DAG2 leads to the activation of the AMPK signaling pathway, a central component of cellular regulation. AMPK functions as a metabolic regulator. When active, it shifts the cell's focus toward energy efficiency and waste management, restoring health to dysfunctional cells. This pathway reduces toxic lipid droplet accumulation and accelerates autophagy, the internal process where cells break down and recycle damaged components. These heat maps show a coordinated increase in activity for genes tied to mitochondrial health, metabolism, and microglial activation. By shifting the metabolic state of the brain, the treatment provides the energy required for cells to clear disease-related toxins. Addressing the metabolic dysfunction of the entire cellular network provided the necessary resources for the brain to overcome the disease's physical progression. These findings suggest that curing neurodegeneration may not require the complex task of replacing millions of lost neurons through transplantation. Instead, a pharmaceutical approach that suppresses the PRAKG2 gene or directly activates the AMPK pathway could enable the brain to use its own existing support cells to clear amyloid buildup. Tracing the path from recovered mouse memory to a single regulatory gene has provided a specific, evidence-based strategy for the future of Alzheimer's therapy.